Then let's go to the topics about today's the communication infections. So then that's why everyone try to communicate, try to reflect what they're trying to talk and controls by some things. So if people communicate, of course we use voice, we use message, we use text, via application, via telephone. Sometimes from your posture, from your face, all the thing can can tell the story about yourself inside. You cannot fake for long, for unless of your life that you are what you are. Then the way you communicate will reflect yourself. So let's see the way of communication is from cell to cell. So look at the host and something in the host like bacteria, virus. Then all the things can share each other with what they want, but we never know what we never hear that. We never heard what they talk each others, but actually they can talk to each other because they have some matchings of the gene. You know, in the human genome, there is a similar of the gene set, at least 37% of the human genome that's quite similar to the bacteria. That's why this is one of the evidence that can prove that need we have some relations with all of the small living in yourself, and also the vertebrate. They have some unique genome set, like 13%. So that's why this is the overall proportions of the gene that share to specifics to in each group, like primate, animals, vertebrates, eukaryote, and bacteria. So this is one of the secret that why the bacteria they have like some chemicals and enzyme, but how come we still have receptor for them? Because we from another world, we from another planet, but bacteria can produce something like toxin. The toxins recognize some things on the human cell. That's why this is a relations between host and bacteria. So after that, what reflects from the communication? They need to make some function beneath the homeostasis between host and the reservoir and the parasite. The language is not like English Thai language. The language between host and the small living cell is about the chemical substance. For example, like lipopolysaccharide, short chain fatty acid, hormone, epinephrine, indole. This kind of the thing is a amount of the chemical members that every day they use to communicate each others. So let's say this one of the secret that inside out secret. Inside out, what happening to your stomach is reflect your behavior, your think, your mind, your ideas. Why this kind of the insects need to consume a lot of cellulose because of the microbiota order this thing. This is inside out. And also sometimes the bacteria can communicate and orders their life to do something phenomenal. For example, in the normal condition, yeast cell, there's habit normally in yeast form, but something happened abnormal in the stomach or about the immunities, like impair of some immunities. There is some chemicals that order the yeast cell, the normal yeast cells, become um, become like pseudo membrane or make like a pseudo hyphae. This one. This is from some chemical signaling that order that kind of the yeast. Like in terms of the candidas, you know the candidas, you got candida in yourself, but all of the candidas never be hypha form in healthy human. But in terms of the immunocompromised, the the yeast convert their physiologies under like heat shock protein or something, and they convert themselves from yeast form to hypha form. This is one kind of the response of the cell once they recognizing something's abnormal. The thing happen under these pictures. Yeah. First thing, the signaling molecules may be carbohydrate, protein, like lipoprotein, glycopeptide or something. These will recognized by the receptor on the cell membrane, and then the receptor tells something like rely to another and change the molecules until they're telling this thing to the DNA of the host. After that, the host respond by cell response. So all the things in your bodies use that phenomenon every day. They started from signaling cells, right? The signaling cell produces extracellular signal and recognizes by receptor. And all the receptors after consequently secreted in intracellular signaling molecules, and then go to the receptor proteins of the target cell. So in this pathway, it's happening in 4 classes. The first one is endocrine. So in terms of endocrine, you can imagine it's about like thyroid hormone, like sex hormone that's produced from your brain, not your organ. From your brain, order something, you feeling something, that feeling secreted in the blood. The cell that secreted some kind of the hormone released to blood circulation, like the thyroid hormones that orders to feel exciting, to convert something in your heart cell, something. And then the receptor is quite far away from the endocrine cell. They need transport. So this kind we call endocrine. The second one is paracline. Para is beside, is nearby. It's something the cells order the cell nearby them, like T cell, T helper cell, tell plasma cell, to do something to convert the B cell to be plasma cell. We call differentiation, right? And the neuron use neuronal signaling. So this one have a little bit complicated pathways. They use we call neurotransmitters. These neurotransmitters will gathering the end of the synapse, and they use this kind of these chemicals to tell the adjacent cell to do something. Finally, at the beginnings of the birth cell, when you was with your mothers in during pregnancy, so people understand why the the, the cell we starting from one cell and replicate exponentially, and in every single cell they know their own function and become the embryos, and all the embryos can convert the functions of the cell to be head, hand, brain, stomach, lung. These things, they communicate via contact dependent signaling, especially in terms of the delta cells. So behind the slide, I'm sure this is a signaling molecules, maybe protein, peptide, amino acids, nucleotide, or even gas. They could be signaling like nitric oxide. This is an example, so I'm sorry for this one. So, but basically, it's about four class of the communication. Like this one is hormone, like adrenaline, cortisol, insulin, testosterone. So it's a kind of the hormone signaling molecule. This is the originals and is the chemical characteristic and the function. So it's nothing to do about this one because maybe it's quite far away from microbiologists, but I just give you a very simple sample on this one. And the locals mediators. And this is a samples, especially histamine. So once the histamine releases, what's happening? You feel uncomfortable and itchy. Or previous conditions make it from vasodilation, right? Accumulation of some kind of the cell, especially the mast cells, and make you very strong itchy. This is one kind of the response. At just very local. So that's sometimes we call local mediator because they happen at local because it's cell-to-cell communication nearby. Also, the neurotransmitters. This one is very famous with these two chemicals, acetylcholine and the GABA. And also the delta cell that's responsible for the cell change during that try to produce from the young cell to adult cells. So let's see what's going on inside. Like this one is the neurotransmitter cells. This is the samples that sometimes the chemical signaling tell the function of the cell, but it depends on what type of cell they try to order. And the function is different from the type of the cell. So we can see the cells that were ordered from acetylcholine. If the heart cell and saliva cell and skeletal cells, this is the same signaling molecule, but the different function. Of course, is opposite from heart and skeletal cell. Once the skeletal cells get the receptors from the neurotransmitter, this one's gonna make the muscle contraction. On the other hand, we found once they attach to the heart cell that become muscle dilatation. So that's why once you order to your friends or your employees in the same orders, maybe the result got different thing. That depends on what you try, whom you try to order. And The next, so I'm trying to show the function of the cell that relied on what type and degree of the orders from one cell. So if I just kindly order to you, maybe you maybe think, oh, okay, I'm very kind and generous, so you're not gonna do that suddenly. But if I push something strong and everyone told you at the same time, at the same story, so maybe once I order something, so in the morning, so in the evening you can finish that. Then it depends on how strong and what type of the signaling that try to push in the cells, and the cell will respond that relied on the cascades or the set of the signaling molecule set. Yes, from the cascades, it's very easy to understand that this is the outside of the cell, this one, and this is inside of the cell. From the outside of the cell get the signaling molecule here that totally match, be recognized by the receptor. And the receptor telling something on the intracellular molecules here and produces some signaling. There is a lot of cascade here, and the signaling can be amplified. Amplified, that means maybe very light sound of the signalings can be amplified like a high voice, big voice, and the cells can do respectively with a lot of respect to that kind of small voice sound, because they have the mechanisms to amplify the signaling here and then so let this kind of signalings do their function. This is the examples. You know about the botulinum toxin. The people who consume some kind of the fermented vegetables is several years in Thailand at Nan province. People at the villages take they took young bamboo fermented food, and yeah, half of them went to the hospital from the symptom of they cannot breathe, they cannot survive because of the muscle the what we call the pulmonary muscles and the thoracic muscles cannot contract. They just keep dilated over time. So at that time, the doctors need to maintain their breath with I don't know, the succinators or something. Yeah, and that kind of thing maintains the patients can still survive. At that time, we can explain how the bacteria try to order something to the body that so in the normal conditions, the neurotransmitter releases from the synaptic vesicles at the end of the synaptic gap. Inside enriched with the acetylcholine, and the green acetylcholine, once they release it at the synaptic gap, they have the target to contract their muscles. This is the normal conditions. But in terms of the botulinum toxin, this kind of toxin block the releasing of the synaptic vesicles at the synaptic gap. That's why the green acetylcholine cannot release it at the gap, and the receptor don't know what they need to do. So during breathing, they cannot do it. They keep maintain dilatation on the muscle. That's why it's very crisis period. This is a kind of the botulinum toxin. But in terms of tetanus toxin, it is the same way, but it's in the opposite action. So normal in terms of the synapse, they need to control when the neuron orders to the muscles to contract. Then the neuron need to tell, okay, it's a time to stop. So by releasing this kind of the neurotransmitter, we call inhibitory intraneuron at this one. So this one will tell, okay, you stop releasing acetylcholine, otherwise the muscle will always contract. Then in terms of the tetanus toxin, this one will block. Yeah, okay. This one will be blocked by the toxin. That's why the muscle still contracts always, because this kind of the toxin cannot work balancing each other, contract and dilate, contract and dilate. But anyway, I love botulinum toxin. It's good for me. These are overviews of the communication between cell to cell, bacteria to host. So just listen to bacteria, what's happening, so why the bacteria can talk, and what is the mechanism that we can explain during 2 hours. So this already is half hours. Okay. Long times ago, not almost, over 100 years ago. So there is a good word that Professor Smith said. I think that multiples of the bacteria are stronger than a few, and thus unions are able to overcome obstacles too great for the field. So this is the very common things when compared to what Thai people believe about the unity. So one cannot work that well. We need a team. We need unity. We need respectful. This is very important for Thai peoples. So once this guy, like just maybe 2 or 3 peoples, this kind of the student not be able to fight to another schoolboy. But whenever they're gathering together, maybe 10 people, 10 students, 20 students, they're ready to fight. And they feel very strong in their mind, and they ask to fight to all the thing around. So like this one. The post-tester as well. So small group has no that powerful. Once we got a lot of post-tester, 10,000, 100,000, a million. Yeah, the signaling is quite different. So once you gathering in front of the faculties, maybe 20 people to ask for something, no one's gonna listen to you. But if you have totally maybe half of the faculty members gathering around the faculties to request some things from the executive staff, of course they're gonna listen to you. So fight for it. I pray for it นะ I pray for Myanmar นะ
ค่ะอาจารย์
Okay, this is a kind of the gathering power. So have a guess, what is it? Is it a satellite picture? Satellite, the picture from the satellite space from out of the world. They took the picture and they got this kind of picture. At first, American thought there's gonna be attacks from Russian because they thought this is the big thing, the large of one 155th mile large. Maybe it's a kind of the weapon that try to maybe destroy the countries. So the scientists go to that areas and pick up some samples. Finally, they discovered that kind of areas just bacteria gathering together, and they produce luminescence signaling to each others along 150 miles. We call this phenomenon as the Milky Sea, like ทางช้างเผือกเนาะ So this is a strong appearance that created from very tiny small living. So from this picture, they reflect something when they try to make pathogenicity in the bodies. So if one bacteria cannot do that, if the population is large enough, they can create something like virulent factors like microbial resistance, the other thing, biofilm. This is a powerful of the people. Once the gatherings, they can create something up there. This is from very junior high school phenomenon, but the below picture, this is from our faculty's gatherings. We do, yeah, a little bit, yeah, I'm proud of it. So the purpose of the communications during the cell to cell, especially the bacterial cell to the bacterial cell or the bacterial cell to the host cells. First, they try to produce the bioluminescence. Is the boundaries, which is the area of the bacteria. They try to create this kind of thing. The second, to create a virulent protein enzyme. They try to produce something escape that's from unwanted or crisis conditions, such as antibiotic use. That's why they need to produce antibiotics by their own, like in terms of the penicillin. That they got some penicillium. Once they grow with bacteria, they produces penicillin to kill the bacteria. This is a crisis condition. And sometimes they produces the biofilm that we call extracellular polysaccharide matrix. A lot of this one will accumulate around the bacteria and become like house of the bacteria. And also they created the communications for talking each other to make a hierarchy of the bacteria. Who should be the main, who should be minors, like who should be wife, who should be distressed or something. So then look at the biofilm or the communication that created the biofilm. There is also bad and good outcome. The bad thing is, of course, the submarine machine don't like biofilm because they affect severely destroy of the machine. That's why the Army US, the US Army invest, I think, over $1,000 million to clean up all the equipment and weapons of them. This is from bacterial communication, from especially the biofilm. On the other ways, the biofilm in your mouth is very close to you, right? Every mornings you feel something slum, the slummy in your mouth, and then you need to clean up every month, every mornings. That is one kind of the biofilm, because you let it glow at night, and during your sleeping you spend 8 hours or 9 hours for sleeping. Is much enough time to produce it. Anyways, in terms of the symbiosis, once they work together, like bacteria to bacteria, the bacteria can make like symbiosis or communities and can produce something very beneficial. For example, like petroleum, gas, oil. This thing also is the best products from communications, but it spend a lot of time for that. So let's see the mechanism. That's very interesting. That, as I say, once yourself, you are in the room and you talk to yourself, the government never listened to you, right? Then you need something, you need some people to agree with you. Then ask some more people to come to join the room. So the language that you try to communicate is one kind of the chemical receptor, the chemical signalings. You talk much, but just a few people listen to you. So the phenomenals of the communication, or the outcome from communication, is not that work. Then what you need is a lot more people to attend in the rooms over the threshold of the population. You can notice about the BPOs hemolyticus or BPO correlase. They produce the fluorescence in the shrimp field, in the shrimp field water. So, but if one isolate cannot produce that kind of the green luminescent on the water, but if they have enough population, they happen to this. But the question is how high of the threshold can create that kind of the outcome. Some species, maybe one colony, some species over the maybe 10 colonies. So this is the outcome. Okay, they have luminescence themselves and become green. That means, okay, all the conversation or the communication already work. Something responds after they try to push, something appears on that condition. So all the thing we call quorum sensing. The quorum means like when we meeting around, like a round table meeting, we talk to each other, we call this is a quorum. We make a quorum. The sensing, of course, sensing is sensing. Sensing is something, okay, touching or something, okay, resulting, fruitful or something. This one. The quorum sensing in the bacteria. Yeah, we can notice from the deep sea animals, they produces some fluorescence by themselves. Sometimes that's from chemicals like nitrics and sulfide, and some kind of the light they produce from bacteria. So this is the structure of the quorum sensing system. So it's can promoting some kind of the behavior in the population. So in the area that they try to produce some virulent factors, all the cells need to produce the same way of the virulent factors. If we consider one of the bacteria, like they try to produce at least 10 virulent factors. Normally, the bacteria not gonna produce all the 10 poison or the 10 enzyme at the same time. No, they're gonna produce just one or two enzyme at the particular times or the episode of infection, and then they're gonna think about the next plan. So most of the case, for example, like Staphylococcus, they think about how to attach to the membrane first. Then they need to think about the necessary protein to attach to the cell first. And after they can attach to the cell, they want to colonize. They need something to produce the colonizes protein. After that, if they get enough members, they can produce the enzyme step by step. Like in this become like a institute culture. And then under the quorum sensing, it sounds like democracy because they need to follow the main populations in that particular areas. If okay, all the people want to go to that way, we cannot deny. We need to accept this is democracy to facilitate the gene expressions in the same way. And then that need to create it under the communications by the molecule, the specific molecule. So this is the advantage of the lifestyle. Once the bacteria choose the quorum sensing system, they choose for enhanced access to the nutrients. For example, the bacteria try to produce some receptors to the folate. The bacteria can contain a lot of folates to maintain themselves. But they need to talk each other. Okay, we need to create a siderophore to the outside of the cell, and they want to obtain the folates. Or they need specific environmental needs. They need their territories. They need to build up some boundaries, just only their communication. For example, like probiotic, right? Lactobacillus, they need to produce a lot of the acids, acidified condition. The acidified conditions will get rid of some kind of the alkali bacteria. And the quorum sensing has very benefits to defend themselves from some other competitors, like they produce some penicillin, they produce some bacteriocin, or some like antibiotics by themselves. As I ever told you about the resistant gene, actually we have found like in the era of mammals, that at that time we discovered the kind of antimicrobial resistant gene already, because the bacteria can produce some bacterial defense protein to each other in nature. So that's why this defense actually is from quorum sensing. And also the build up the colony escape. That means sometimes the colony full of the population. That's why they need to jump to another territories that's become expand their population around. This is a benefit of the quorum sensing. Let's see the cartoon that
It
can
would be of
explain.
little value for just a few bacterial cells to produce a gene product, such as an enzyme or virulence protein, because the concentration of the protein would be too low to be effective. Quorum sensing is a type of regulatory process that ensures there is a sufficient cell density before a specific gene product is made. This process allows bacteria to increase in numbers before starting to produce a particular gene product, such as an extracellular enzyme or a virulence protein. Each species that employs quorum sensing produces a small signal molecule. In gram-negative bacteria, the signal molecule is an acyl homoserine lactone. When the acyl homoserine lactone signal molecule reaches a threshold concentration, it binds to and activates a regulatory protein, which then binds to a specific site on the DNA. The binding of this regulatory protein transcription activator results in production of the specific quorum-dependent protein, as well as more enzymes to make the acyl homoserine lactone. Quorum sensing in gram-positive bacteria involves a different type of signal molecule. A precursor oligopeptide is cleaved into functional signal molecules of 10 to 20 amino acids. These molecules are actively transported out of the cell through a special transporter protein. When the signal oligopeptides reach a threshold concentration on the outside of the cell, they are detected by a sensor protein on the surface of the cell. When the oligopeptide reacts with the sensor protein, the protein becomes phosphorylated on the inside of the cell membrane. The phosphate is then transferred to a response regulator protein, which allows it to bind to a specific site on the DNA. This binding results in alteration in the transcription of target genes. Quorum-dependent proteins such as specific virulence factors are produced.
This gone a little bit virtual than what I said before, but anyways, we're gonna study about the signaling molecules. But in these cartoons, there is two groups, right? The gram-positive and gram-negative. The gram-negative that relied on one kind of the molecule we call autoinducers by acyl homoserine lactone. They dump out and that kind can absorb into the cell. But in terms of the gram-positive, they use oligopeptide and they produce and secreted to outside, but they didn't come inside, but they just activate on the membrane. And then the membrane can tearing something in the cell by phosphorylation. Let's see in the detail. This is the structures of the quorum sensings of the cell. So let's see the cell. The cell needs to produce the signaling molecules by synthesizing the molecules. Of course, the synthesizing of this kind of molecule is from the DNA. In some particular areas can produce some kind of this thing. And then they diffuse via the membranes to the outside of the cell, but they never leave from the cell. They still gather around the cell. And after that, once the concentrations of the signaling molecule is high enough, then these kind of the molecules will be reabsorbed into the cell, and they switch off some things about the receptor under the receptors, and then this kind of protein inside will bind to the promoters to allow the cells read this kind of DNA and produce some protein, become gene expression. This is the outcome of the gene expression. The phenomena may appear like bacterial attachment, motility, biofilm, vitamin synthesis, or oxidative stress response. Then let's see this is the the structures of the quorum sensing. First, we need to have microorganism, right? This is like free living. You need some kind living, and then the the bacteria got the gene that have the functions for creating this kind of the cascade. So this kind of thing should be like a set of the technologies we call the technological tool of the bacteria. And then the autoinducer is very important because the bacteria communicate mainly via autoinducer. All of the signaling molecule we call autoinducer respond in your mind. This is very important. When you want to study about the communications or how to reduce the virulent factor, many of the researchers focusing on autoinducers in terms of the regulation or production. Then the autoinducer equal to the media or signal. And then after they produce the media, they need some kind of the membrane round to accept that kind of the media inside of the cell, right? That's equal to the cell have antenna or connecting box from the communication. So after that, the cell need to screen what kind of the enzyme protein need to produce at that episode. So we have sensor department like in the cell, and also not just only one kind of autoinducers. The bacteria live like niche communities. In the communities, bacteria may find some other bacteria. So they need to talk to other bacteria as well, not just only themselves. So once they talk to other bacteria, they need another signaling not equal to the same intraspecies signaling. We call not intraspecies, it's interspecies. We call auto-inducer 2. So like, okay, you use the signaling from the AIS company. Of course, you can call to detect companies, even different companies, but we need to communicate. So in nature, bacteria may create some kind of some other auto-inducers to talk and to make balancings in their niche community. The signaling, this is a sample of the signaling. Basically, in terms of the bacteria positive, gram-positive bacteria, you see this is the peptide. The peptide is different from lactone. Peptide is peptide. Peptide is comprised from amino acid, right? So the long and the type of the amino acids will convert to be several kinds of the peptide and the protein. So in terms of the quorum sensing signaling for bacteria, at least 10 to 12 or 10 to 20 amino acids will link each other and become autoinducers of the gram-positive bacteria. In terms of the gram-negative bacteria, this kind of the quorum sensing is not peptide, but is lactone. You know what is the lactone? What kind of the chemical, the basic chemical? The lactone is not carbohydrate, nah? Lactone is ester, like sugar ester. It's not pure carbohydrate. So the full stage of the AHL stands for acylhomoserine lactone. And the autoinducer too is this depend on what kind of the interspecies communication from gram positive to positive, positive to negative, or negative to positive. It depends on that thing. And the type of the Acils homocellulactone is depend on species of the bacteria. Let's see this picture. The lactone, actually lactone is a cyclic carbolic ester. So this is the cyclic one. It's the backbone of the lactone. But the differentiating thing is about the R group behind this one. This makes differentiate in each unique species. But anyways, to produce it, the autoinducers, the bacteria need to consume or need fermentation until they can produce enough ATP or energies to produce it. Then this reflects something about the gram-positive bacteria may need a lot of nutrients to create the autoinducers for making some communication. But in terms of the gram-negative, it sounds like intermediate cause of metabolic cause. But in terms of the talk to each other between the species, then they use just autoinducer to maybe they just automatically talk to each other because they just reflect something, but don't need some like appearance or some change from communication. Okay. So in terms of A does reflect the gram-negative bacteria is the principles of the communication phenomena. First thing from the cartoon, right? They have the gene that represented luciferase protein, this one, and Lux I produces Acils homocellulactone. They jump out from the cell to outside, and then until over the threshold of the chemicals gathering around the bacteria, AHL or acyl homocellulactones can reabsorb into the cell where describe the receptor. And these chemicals will attach to the receptor inside in the cytoplasm. And this one will regulate and orders the gene, the promoter gene, to read and translate from DNA to RNA and RNA to specific protein. This is the classical one for gram-negative bacteria. But anyway, in terms of the gram-positive bacteria, this one origins from produces some of the oligopeptide, right? And the oligopeptide will be cleaved or cleavage until get small enough to release at least 10 to 20 amino acids become oligopeptide, and they release it out to the cell and gathering around. But in terms of the gram-positive not let the oligopeptide reabsorb to the cell, but the oligopeptide just accumulate around and attach to the receptor we call the sensor. We have sensor here. Most of the sensors from sensor kinase is the main of the metabolic chemicals in the body. 70% of the bodies they have sensors kinase, and they switch the molecules behind the sensors by using phosphorylation. Of course, this one will give the phosphate attached to this kind of receptor. And then a phosphate from ATPs, the one phosphate can release it to the regulators, the response regulator here. and then it's ready to attach to the promoters on the DNAs, and created some the phenomena. The C1 is like parallel component signaling, not just only one autoinducers. They need double autoinducers to work together, and then this kind of the things can assure the microorganisms to work in rigid pattern more exactly. It's good for bacteria, even it look very complicated, but it's good because if some institutes decided by just only one people, it's very risky. But it's all of the outcome if it's from considerations of the committee that may be more sure safety and more rigid. I cannot teach you all of the thing of this, the autoinducers that related to the bacteria and the phenotype response. It's not possible. And I don't know about exactly what inside in each unique bacteria. So, but it's just an idea that you can like enhance your knowledge to use in terms of your thesis or your future research about how to study or the basic knowledge, the principles of the quorum sensing. So I'm going to tell you a little bit about the samples of the quorum sensing, at least 4 types of the bacteria, and then I'm going to talk to you about the applications. After once we know about the quorum sensing, what is the benefits for this kind of knowledge? This is the concept of the quorum sensing. When we're talking about the quorum sensing, we're thinking about the aquatic squid. We call bobtail squid, ใช่ไหม Silavit, did you work with bobtail squid, right? About fish, aquatic animals. So who your supervisor? I'm sorry. อาจารย์ชันนรงค์ใช่ไหม what did he assign the thesis topic for you?
Inhibitory biofilm pan extract.
Oh, perfect. Biofilm for which kind of the Faubacterium? Yeah, that is some report about the Faubacterium quorum sensing already. That's good one. Okay. This is not Faubacterium. It's not a Nighttilapia. But this is bobtail squid. The bobtail squid is very classical samples to explain the quorum sensing. So let's see at first I will see. Okay. So one day was born after they hatched from the egg within 8 hours. Let's see. Actually, on this we call anterior appendix from mouth of the bobtail squid, still quite very long, contained with a lot of cereals around this kind of the appendix. Is ready to use for catch the nutrient around. This should be like that, but finally we found something happen on this kind of the appendage is become shortened from 12 until 2 days and until 4 and 5 days later. It's become shortened from long ciliated epithelium until short ciliated back epitheliums. So the scientists very surprised what happening is to this kind of the very poor baby bobtail squid. Oh, these ones were accumulated with a kind of bacteria called Vibrio fischeri. The Vibrio fischeri colonizes around of this on the bacteria, and they destroy the epitheliums and become shortened. And this kind of the shortened is not good for squid. But anyway, the kind of the compensation from nature. Luckily, this kind of the bacteria can give luminescent. So after 5 days of birth, the bobtail squid will have light at around the mouth, and then the planktons will come to the mouth easily. They don't need the ciliated epithelium anymore. But anyway, this phenomenon very interesting because they're thinking about some kind of the deciliated epitheliums in terms of the pathogenicity of some kind of the bacteria in human, especially in terms of the severe coughing of human. So the Vibrio fisheries, they use the mechanisms like the classical mechanism of the quorum sensing in terms of the gram-negative bacteria. They have ability to create a luminescing receptors, inducers, and they created the inducers outside of the cell and come reabsorb to the cell and switch on the promoters and give a lot of the gene expression occurrence here. Biofilm, conjugation, swarming, virulent factor, symbiosis. So more than that, the scientists try to use that kind of model of the bobtail squid that they loss of ciliated epitheliums because of some kind of the toxin. And they realized that kind of toxin is a tracheocytotoxin. Actually, the tracheocytotoxin has the analog structures at the membrane of the bacteria. So interestingly, the bacteria, they have a thin layer of peptidoglycan. If you can memorize that, the structure of the cell. At that areas, they accomplish with the glycan and peptide. The glycan comprising acetylmulamic acid and acetylglucosamic acids, or NAC and NAM. In these structures at the peptidoglycan layers can be themselves become a toxin like tracheotoxin. Once they try to colonize at that areas, they will release this kind of thing at that areas. Or sometimes the bacteria were destroyed by antibiotics. They also created this kind of thing. But in terms of the bobtail squid, of course, the bobtail squid during colonization, some of the bacteria loss of this kind of the structures and releases to the area of the colonization. That's why the ciliated epithelium becomes shortened. This kind of models is totally equal to explain about the damage of the ciliated cells in terms of the Neisseria gonorrhoeas, Haemophilus influenzae, and Shigella Veterinary. Helicobacter pylori เดี๋ยวนะ2 นาที ไฟไม่เข้า โอเค เกือบดับแล้วเนี่ย Right now we go until peptidoglycan can act as autoinducer. So that tiny small molecule can do some things over than your thought. So what's happening? This is maintained about the symbiosis and also the antibiotic resistant inducers and a turn strong of the virulent factor at the same time. That is the gram-negative bacteria. So let's see gram-positive bacteria. That is one classical bacteria is Staphylococcus. Then from the principles, they consider about the concentration of the cell at that area. But in terms of the small members of the Staphylococcus, the cell will take action for attachment and colonization first. In terms of the high members, they're gonna release toxin. So then this is the cascade of the action. First, just attach and colonize. Like first, if you have not that much abilities, okay, you just stay still and can keep survival at that areas. If you're strong enough, yeah, you can do whatever what you want. So by this kind of the mechanisms, so briefly, yeah, of course, this is the membrane. Look, sorry, this is the membrane of the Staphylococcus, and on the membranes, they have the receptor and the releaser here, and they need oligopeptide here. They need to release the autoinducers peptide outside. They need the structure here to to attach and activate the phosphorylation. And then the phosphorylations gonna switch from inactive to active of the intracellular signaling and tell the promoters to read to enhance the concentrations of RNA3 and releases the action, the response. Then let's see. So in terms of the Staphylococcus, we call the mechanisms of the Staphylococcus as the two-component signaling transduction. The classical pathway for gram-positive. Firstly, of course, the Staphylococcus have the gene AGRD gene, maybe aggregative something, and they releases the protein and rearrange the protein and conformations of the protein released via this route and become the autoinducers outside and accumulate all the autoinducers outside until they get past the threshold of the concentration. They attach to the receptor and activate the phosphorylation here and switch on the gene until they gathering the concentrations of RNA3s, and then they ask the cell to do the thing. Then two phenomena happen after the trigger. First thing is in terms of low densities, they will work with Staphylococcus protein A, fibronectin binding protein. this one will be released, this kind of the protein will be firstly releases from the DNAs because they need to attach. That's why the proteins promote the attachment will be predominant. And after the high concentrations, they can release a lot of the virulent genes such as hemolysin gene. That's all of the toxin, all of the bad protein releases one day can strong enough and get a high population enough. So in terms of the trend in microbiology, this paper is very interesting because it answers my hypothesis. One of my question, why the bacteria shoot to resistant instead of the shoot to survive as the wild type. So, and why most of the resistant bacteria not so healthy when compared to wild type bacteria. So this maybe can answer my question that, okay, once in terms of the healthy bacteria, so they have like good immune function. They have nothing to do about the antimicrobial resistant. They can work well with some necessary thing, some necessary protein in terms of the survivals. Then extend this make them strong enough. But on the other side, so once the cells during in the crisis, they have to use the energies and the proteins switch to defend themselves, not for maintenance, but to defend. So ask yourself, if you have to defend to something, okay, you're gonna be weaker than the healthy one, the healthy condition. If you don't have to face something, okay, you spend all with yourself, your life, your growth. But in terms of the conditions of defense, you need to spend a lot of energies. You're gonna get stressed. You're gonna get a lot of virus inside, and then the bacteria, okay, look like it has more abilities to defend the antibiotics. But of course, if you compare about the immunity of the cell with the resistant, sometimes we call mutant bacteria. Of course, the mutant organisms get weaker than wild type because they spend a lot to defend, not for maintenance. The second samples from gram-positive bacteria is Bacillus subtilis. The Bacillus subtilis, there it can choose what kind of the action they need to release it. It's quite the same idea, the same wavelength with the Staphylococcus aureus, but in different system. We have COM system. COM stand for by the complement system. This, of course, the competent system. This system try to maintain the bacteria in healthy condition. Yeah, the compositions of this kind of the system consisting the COM-X is from pheromone. Of course, this is from outside of the bacteria. We have COM-Q. The COM-Q, which one? This one. Okay. And COM-P. COM-P is a membrane-bound receptor. And COM-A here, yeah, is the transcriptional activators of the Bacillus subtilis. The second system that they can work together is the CSF, like colony stimulating factors. So in terms of the COM system, the ultimate goal of this kind of system is to maintain or to make the cell competent. But in terms of the CFS by LAF-C, this kind of system have the final objective to produce the sporulation. So just think about the bacteria. Once they try to make some sporulation, that means maybe in the crisis or in some nutrient loss, or they survive in full nutrients condition. That's why they need to maintain their hierarchy and convert the cell from normal cell to be spore. So let's see the competent and sporulation factors in the low concentration. In the low concentrations, the sporulation flee here, and the competent is from only COM system. Yeah, the COM system that will represent from competent and will be blocked because the colony stimulating spore forming factors will not that release it much, and the system will be blocked this one. But the competent signaling, the competent cascade will be free work and switch the DNA just only to competent. But anyway, in terms of the sporulations in high concentrations, at that times the nutrients become hard to or not enriched at that areas. So the cells try to preserve themselves. They learn to produce the second mechanisms of the cell spore forming and unlock this one. And SPO cascade will be influenced over the COM mechanism. Okay, the next, just go to Pseudomonas aeruginosa. This one is the classical form of the double signaling, and sometimes they work together. The scientists noticed that the Pseudomonas aeruginosa have a lot of abilities to produce too many enzymes at the same time, and why they can produce it at the same time. So here, the auto-inducer of the gram-negative is acyl's homoserine lactone. This kind of the AHL created from LAS and RHL. This is from elastin, and this is from lamnolipid. So two kind of the system will control each others. Let's see how they can control each others. This is the quorum sensing circuit arranged in this kind of the series. So let's see, the first quorum sensing, they're created from elastin cascade. The elastin cascades, they work very well, and the outcome of this cascade to produce the proteinase to virulent factors, the biofilm formation. Once this kind of protein success to produce, they allow the positive feedback to another loop. The loop will go to lamnolipid loop, and the lamnolipid loop will be created some others protein and some others pigment of the pseudomonads, such as pyocyanin. Then if you know this one, so you can imagine before the pseudomonad produce the green colors, firstly they need to build up the biofilm first, and they need to produce all the virulence first, and then this kind of the protein will around the second loop of the circuit can produce the pyocyanin. Too many, nah? The final sample is about Vibrio havii. These Vibrio havii, they use the parallel quorum sensing circuit from quorum sensing, from three mechanisms. The cholera autoinducer one, this one, is the common autoinducer at the first one. They work together with the HAI 1 and AI2 from the nearby gene of the chromosome. So anyway, they use three systems at the same time to produce the luciferase or to produce the luminescent occurrence. Okay, forget about it. So this one will rely on the concentration of the population as well. So in the low populations, luciferase will work very well. But in high population, in the low populations, they're gonna work until almost ending of the target of the gene, but they will replace it by negative feedback. But in terms of the high population, the luciferase will work very well together with once they work with HAI and autoinducer 2 together. This one. But anyway, it's nothing to do of Vibrio havii in terms of the veterinary practice. But just want to for you to have a chance to see about the cascade inside of the cell and how they can work with the circuit of the molecule to the molecules, and how the molecule of the intracellular molecule they work together, maybe positive loop or negative loop. So let's talk about the biofilm formation. So this, as I mentioned before about how we waste money about the biofilm management. This has become one of the worst point when we try to manage during treatment, because even antibiotic work well with bacteria, but sometimes they're facing with the bacteria within biofilm. Just remember that you can see this is planktonic cell, right? The planktonic cell that represent single cell, they can live alone without any protection. The antibiotic can kill planktonic cell directly. So the response level can be represented as the susceptibility test. So once you detect the MIC at 4, so of course at 4 microgram per ml can kill that kind of bacteria. But in terms of the set size cells, no. The sessile cell is very more complicated, and they have their own protection. You cannot use the antibiotic as 4 microgram per ml as the MIC susceptibility tells, but you need to eradicate all of the biofilm before you use the antibiotic. So this is the biofilm structures. So the structure of the biofilms comprises of as a communities of the microbial attached to one surface. And also we have found sometimes the bacteria or yeast can live together if they are living in symbiosis in terms of the commensalisms. And they attach each other. We call the organism in the sessile stage. So let's see, this is my result. We study on the yeast cell, on the skin yeast. At that time, we discovered the both of yeast, Malassezia pachydermatis and Candida parapsilosis. They can live together in dog ear. And we think at first, maybe they're living like competitive symbiosis. So they maybe competition, like competition living. But finally we found they produces the biofilm together and they help each other together. We observed the biofilm productions from singles Malassezia and singles Candida. They're still very low when compared to co-productions of the biofilm from 2 species. And this is the outcome from the antifungal susceptibilities. We can see at least 500 times antimicrobial resistant to the sessile form within biofilm in all conditions. Okay, this is the impact in terms of the infectious biofilm. So we pay attention on the dental plaque, endocarditis from Streptococcus pyogenes, right? And cystic fibrosis in lung, otitis externa, and yeah, of course, we found the problem with the biofilm attached to the catheters, especially urinary catheters, and to the implant organ. So that's why the medical technologies need to prove the establish or the building of the biofilm at the initial stage to prevent the bacteria create their own house in the body. Yeah, people can see this picture a lot about the bacteria, how the bacteria can build themselves and create the extracellular polysaccharide until become house of the bacteria, and some of the bacteria can release it from the house and become another new colonies under the biofilm. This, as I mentioned before, once they produce their own biofilm, the yellow and green that represents for just singles producing biofilm from Candida and Malassezia respectively. But the blue one, very interesting, that once we grow up they together, you can see how fast and high of the productions of the biofilm together. Just represents one is colonized on their ear cavities by both species. It's become quite difficult to treatment by normal antifungal drug. So that's why I give the opinions to pay attention on how to manage the biofilm formations before treat of the antifungals. This has been published in Medical Mycology. So look at the internal structure of the biofilm. There is at least 3 layers of the biofilms. Rely on the functions of the biofilm structures. Of course, they need to build up this kind of the house of the bacteria for unleashable antibiotics outside, right? So that's why the yellow antibiotics will around outside of the probiotics, but they cannot penetrate until the internals of the house. Then this is we call unleashables labels here for unleashables, and inside is a layer of resistant phenotype. All the resistant or persistent bacteria can survive inside here. So that's why if you pick up the biofilm from treated animals, you still found some resistant bacteria or some persistent bacteria from the samples. This is from in the internal layer, in the intermediate layers here. And finally, we have outer micro environment. This kind of zones got nutrients depletions or waste of the product, because this is have the gathering with the old cell inside, and some of the old cell will attach to the membrane with the absorption of the antibiotics from capillary here. So that's why we may see some kind of the dead cell here. But anyway, what the fruitful microorganisms, we pay attention on the middles of the biofilm. But in terms of the chronic wound, the biofilm created on the top of the chronic wound to protect the stem cells from the surface. And not just only antibiotics, they protect stem cells, the bacteria from the immunities, especially the complement and antibodies. And some kind they protect us from the very important leukocyte. And most of the leukocytes can create a lot of damage inside and the oxidative stress. So this one, the biofilm can protect the bacteria from this kind of the enzyme. So look closer to that in terms of so we call sinusitis. The chronic sinusitis, the people who got this kind of sufferings will be suffering maybe most of their life, until the rest of their life maybe, because in the nasal cavity in these areas, the bone will arrange like labyrinth. So this is very complicated, the areas, and it's quite difficult to let the antibiotics go through or all around this kind of area. So that's have much potential to be area of bacterial colonization. And once the bacteria colonize, so even you use the spray, antibiotic spray to your nostrils areas, or giving systemic antibiotic administration, is not possible to destroy the biofilm inside of the labyrinth of the nasal marrow and nasal bone. So one thing in the medicals tool and medicals regimen, they mix some kind of the sugar ester inside to dissolve the ligit of the biofilm inside of the labyrinth here to become unsolid, like let it softer and maybe can release it from that cavities by using the xylitol. This kind of the chemicals you may familiar with the gum, right? The gum mixed with xylitol and sucrose. Yeah, if you don't brush your teeth in the mornings, you may use the gum, that kind of the gum, to clean up your mouth. So in terms of emergency now. So that is for the pathogenesis of direct bacterial to bacterial and bacterial to the host cells. And this kind, I jump to what the host can destroy themselves by their response. So let's see in terms of the stress. In terms of the stress, the cascade of hypothalamic, pituitaries, and adrenals exist. That once you get stress from your brain, what you think is gonna respond here and releasing to the target until to the adrenal glands cortex. And what happening from this one? From hypothalamus till until the adrenal glands. The adrenal glands always release the steroid into your bodies. On the other ways, the systemic nervous system, once you get stressed, they're also releasing some kind of the chemicals we call the signal of the stress hormone, that is epinephrine and norepinephrine. So they all work together. So people know about the steroid. Steroid can inactivate the leukocyte, and norepinephrine and epinephrine is the hormone of stress. So what happens to the quorum sensing of the bacteria? Because the epinephrine and norepinephrine has very similar structures or analogs to the autoinducer of the bacteria. Then if you get stressed, bacteria know, okay, you are stressed right now. So this is what we call is inter-kingdom communication. So bacteria can respond after we think. So even, okay, you not intend to think about this, but naturally, if you, your characters always think bad, you always think, get stressed. Yeah, the bacteria inside of your stomach know about this condition. And then epinephrine, your own epinephrine and norepinephrine will attach to the receptor of the cell. And what's going on after that? The cells will get high densities. Your normal flora is not in homeostasis, but especially Salmonella will get high number. These happenings have been published for Salmonella in pig already. So this is the food poisoning caused by Salmonella and EHEC. That this kind of the papers tell the story related to the auto-inducer trees we call misuse signal. This from the stress of the host by epinephrine and releasing of the norepinephrine. This can produce and promote and regulate the bacteria to produce the sinusitis and to enhance or to make the bacteria secreted or much more virulent factor during stress of the host. Then just think about this one. This one is very good samples. It's nothing to do what you facing in front of you because you cannot control them. But it's about what you think about that. So in terms of veterinary practice, so most of the cases they are study about the Staphylococcus pseudomonas salmonella and some of the vet pathogens like Leptospira also. We have study about the quorum sensing system, Flavobacteriums also. Good luck. And the futures of the quorum sensing. The research go to find out some signaling molecule detections at the beginning of the infection. They need to observe and try to protect the bodies from quorum sensing. And then they try to pushing some chemicals to treatment of the quorum sensing by maybe interference of the signals between cell to cell or destroy the quorum sensing. No, destroy the biofilm directly. Okay, some of the study already success to tell some kind of the biomarker to indicate the onset of the pathogenesis after infect to some particular bacteria, and they can follow up the infection by using some biomarkers. Of course, that kind of biomarker is auto-inducer. So this is the sample of the quorum sensing inhibitions they use as the therapeutic tool. So in terms of the bacillus, the researcher used the acetylhomoserine lactanase as the enzyme to eradicate autoinducer directly. Interestingly, luckily the researcher found some benefits for macrolide like tylosin, erythromycin. So even this kind of the antibiotic cannot kill the bacteria, but luckily they can interfere the bacterial pathogenesis by interfere the signaling of the bacteria. And some others, the supplement to inhibit the quorum sensing. So almost 2 hours. Okay. This is very concrete evidence. They use the one kind of the sugar ester coated on the glass surface and polystyrene like urinary catheters to prevent the biofilm during the times of incubation. Like the patients, we use the IV catheter, right? The catheter that coated with the sugar ester, and this one can prevent the accumulations of some kind of the bacteria or yeast during the maintenance of the treatment. Then also this kind of the sample that they try to created some immunotherapy antibodies that against to the quorum sensing. This is the easiest cyclic carbolic ester, right? This is acetylhomoserine lactone, and this kind of the homoserine lactone can be destroyed by the squaric monoester. This is a kind of the heptane, the synthetic heptane in the in vitro only. So this kind of the heptane can create this kind of the ring and can destroy the auto-inducer structures and become inactive. Finally, they try to use the bacteriophage to coat on the catheters to reduce the chance of the bacterial colonization and building up the biofilm. Do you think this is possible? This is catheters, and they called it some bacteriophage here. Yeah, very clever to use the hydrogels contain bacteriophage and ready to kill the bacteria once they try to colonize here. What do you think in terms of the application? Yeah, actually, look at this one. So how many bacteriophage type you need to prepare for catheter coat? Because bacteriophage has their own properties to be very specific to the species or sometimes to the strain. So just one bacteriophage can eradicate just one kind of the bacteria or the strain. We cannot choose what is the bacteria can come. So what is the strategy? I don't know, but is very interesting if you want to study about the quorum sensing and how to apply in terms of the quorum sensing to your thesis or to your future research.